Okay, so let's move on to the synapse. The nervous system works because we're taking information from one neuron to another neuron. So in this case, our synapse is going to be the axon terminal of one neuron to the dendrites or the cell body of another neuron. Before, when we talked about the skeletal muscle system, we were talking about the axon terminal from one neuron onto a muscle itself. But here we're talking about typically from one neuron to another neuron or from one neuron to an effector cell, such as an organ or a gland. So when we're talking about the synapse between two different neurons, we have, let me just draw it here. This area here would be our synapse. The initial neuron over here that's actually sending the impulses towards the axon terminal, we call the presynaptic neuron. The one who's actually receiving the signal or receiving the information is the postsynaptic neuron down here. We'll talk a lot more about this in the last exam unit. So for lecture exam 6, when we talk about two-chain neuron systems and how these function. Um, but again, postsynaptic neuron would be the one receiving the signal, but it doesn't just have to be a neuron. We could also be talking about a muscle or a gland that could also be part of the synapse itself. So we have a couple different types of connections, and basically their name is self-explanatory. So if we're talking about axodendritic, we're talking about the axon of one neuron synapsing on the dendrite of another. If we're talking about axosomatic, we're talking about the axon of one neuron synapsing with the cell body or the soma of another neuron. Or we can have axoaxonal, although less common, where we have the axon of one neuron synapsing with the axon of another one. And when we talk about the actual synapse itself, there's a couple different types. We have our chemical synapse, which is basically the use of a chemical in order to transmit the signal from one neuron to the other. So that would be an example of, say, acetylcholine. We're talking about electrical synapses. We're talking about the movement of ions through gap junctions between cells. So here's just an image. These are the three that you have to know for your lab exam. So here we have the axon of one neuron here in green synapsing with the dendrites of this main neuron shown in this picture. So we call that an axodendritic synapse. Down here we have the axon of one blue neuron synapsing with the axon of this neuron. So in that case we call it axoaxonal. Over here we have the axon of a one neuron synapsing with the cell body, which is also called the soma. So we call this an axosomatic synapse. So we're predominantly going to focus on chemical synapses, although just know we can have chemical, which we'll touch on in a little bit. But the most common type is a chemical synapse, meaning that we send the signal across the synapse in the form of a neurotransmitter. So it's composed of two different parts, and this should also be similar to what we learned in the skeletal system. So the axon terminal of the presynaptic neuron, so the one sending the signal, is going to contain synaptic vesicles filled with neurotransmitter. The receptor region on the postsynaptic neuron's membrane, so the receptor region would either be somewhere on the dendrites or the cell body, is going to receive the neurotransmitter. And there's going to be a space between the axon terminal and the receptor region, and that's the area that we call the synaptic cleft. So basically what's happening is we're taking an electrical impulse and we're changing it to a chemical signal as it crosses the synapse, and then we're converting it back to an electrical signal once we finally have that, that action potential generated in the axon itself. So a little bit more about chemical synapses. We have transmission across the synaptic cleft, and that's going to prevent nerve impulses from directly passing from one neuron to the next. So it's going to actually slow it down just a little bit. It's a chemical event as opposed to an electrical one because we're using a chemical in the form of a neurotransmitter. So it's going to depend on releasing that neurotransmitter, the ability for that neurotransmitter to diffuse across the synaptic cleft, and the ability for that neurotransmitter to bind to the receptor on the postsynaptic membrane. The whole point of transmission across the synaptic cleft is that we're ensuring unidirectional communication between neurons. So we have 6 steps involved, and again, these should remind you of what happened when we talked about the neuromuscular junction. So the first step is that is going to begin with neurotransmission when we have the action potential arriving to the axon terminal of the presynaptic neuron. So that's what it's showing you here. We have an action potential that was generated in the presynaptic neuron. We've opened all of these voltage-gated sodium channels, and eventually we're going to change the voltage down in the axon terminal. So that depolarization of the membrane caused by the action potential is going to cause the voltage-gated calcium channels to open, at which point we're going to have calcium come in and enter the terminal. And if you remember what the function of calcium influx into the axon terminal is from the neuromuscular junction, it's the same thing as it is here. So we have calcium entering in down its electrical chemical gradient from the extracellular fluid into the axon terminal itself. So that's what step 2 is showing you. Now we've opened these calcium voltage gated channels and calcium is coming into the cell. And the third step, calcium entry into the cell, is going to cause the synaptic vesicles that are filled with neurotransmitter to empty their contents into the synaptic cleft. I don't want you to worry about this. I think that's too much information for AMP1. Just understand that once calcium comes in, the synaptic vesicles are going to fuse with the plasma membrane and exocytose the neurotransmitter into the synaptic cleft. So that's step 3 showing you here where they've fused and now they're releasing their neurotransmitter into that fluid. The 4th step is where we're going to have the neurotransmitter diffuse across the synaptic cleft and bind to its receptor on the postsynaptic membrane. And most of these receptors are chemically gated ion channels, just like we saw with the acetylcholine receptor. So step 4, our neurotransmitter is going to come in and bind to a chemically gated ion channel on the postsynaptic membrane. Again, they're showing you a postsynaptic neuron, but it could also be the membrane of a muscle or a gland as well. So step 5, binding of neurotransmitter is going to open ion channels, and that's going to create a graded potential. So this is something different than from the neuromuscular junction, because once we had our neurotransmitter binding to our muscle cell, that generated an action potential. When we're talking about neurons, we have graded potentials initially in a receptive region, and then if we get enough graded potentials that are stimulatory, then we can go on and generate an action potential. So we're going to have our neurotransmitter bind to the receptor, which are chemically gated ion channels, and they're going to create some sort of graded potential that can dissipate over the course of a distance. These graded potentials could be excitatory or inhibitory. We'll talk about in a little bit what an excitatory graded potential is. That's going to be one that's going to bring the resting membrane potential closer to that threshold of negative 55 millivolts. An inhibitory graded potential would be one that's going to bring the membrane potential more negative than the resting membrane potential. So say like negative 70 or negative 80 millivolts, that would be an inhibitory sort of graded potential. But again, the big difference here compared to what we saw with skeletal muscle is that these neurotransmitters can cause either depolarization or hyperpolarization of the cell because they're creating graded potentials. And the skeletal muscle system, they were always excitatory and they always were depolarizing signals. So that's what it's showing you here. Our neurotransmitter is now binding to its receptor and we have movement of ions. The last step is we have to degrade the neurotransmitter there so we don't continue to send the signal. We have a couple different ways in which we can get rid of the neurotransmitter. Just like we saw in muscle, there could be an enzyme there. So like we had acetylcholine esterase present in the synaptic cleft, we can have other enzymes there ready to degrade them. The third or second way is that the astrocytes, remember one of the supporting cells in your central nervous system, can reuptake that neurotransmitter to either recycle it or do something else with it. or the axon terminal itself that originally sent out or released the neurotransmitter can also reuptake the neurotransmitter. And the third possible option is that it simply just diffuses away from the synaptic cleft. So that's what it's showing you here. Three possible ways we can get rid of that neurotransmitter that was bound here. We can reuptake it into the axon terminal. We could have an enzyme present there to degrade it, or it can just simply float away and no longer be present in the synaptic cleft itself. The last thing we need to talk about when we talk about the synapse is something called synaptic delay. And this is the time it takes for that neurotransmitter to actually be released from the axon terminal to diffusing across the synapse to binding to its receptor. And that time can vary depending on the synapse itself. So because of that, we say that the synaptic delay, which is these three events, is the rate limiting step of neural transmission. So it is the slowest step and what we call the rate limiting step. And this is just one slide. We just talked about chemical synapses, which require some sort of chemical in order to bind a receptor. I just want to briefly touch on what an electrical synapse is. So these are much less common, and I mentioned previously the adjacent cells are going to be joined by gap junctions, which are basically going to allow ions to move in between the different cells, which are going to change the polarity of the membrane, and that's what's going to cause these cells to be electrically coupled. Other than that, you don't really need to know too much about electrical synapse.